Hey there, welcome back for another deep dive with us. Looks like we're getting into some seriously detailed stuff this time around. You've brought us a ton of information about GLP protocols and standard operating procedures. SOPs, right? Those are important in lab studies from what I remember.
Yeah, you're right on the money there. And you've given us some really interesting sources to work with, too. I mean, we've got everything from those official regulatory documents like CFR Titles 21 and 58, which, lay down the law, so to speak, all the way to these super practical YouTube videos that break down GMP and GLP. Plus, we've even got some excerpts from books on pharmaceutical manufacturing and just quality management in general. It's a really fascinating mix.
It is. So what we really want to get into today is how all these detailed protocols and procedures, I mean, they get pretty intense, actually work in practice. I'm especially interested in how they guarantee that experiments are done the same way every time, consistently, and that the data coming out of labs is solid and reliable. And that's especially crucial in fields where, like, messing up even a little bit can have huge consequences.
Absolutely. And for you listening in, this isn't just some theoretical exercise, right? This is about understanding the very foundation of how we create trustworthy scientific data, particularly in critical areas like developing new drugs and making sure those medicines are manufactured safely and effectively. So we're going to break down these potentially complex, maybe even intimidating topics and show you why they matter so much.
Okay, so let's jump right in. Why are these GLP protocols and SOPs so incredibly detailed? Like, seriously, when you first lay eyes on them, it could be a little overwhelming, right?
Oh, I totally get it. They can be a bit much at first glance. But there's a very, very good reason for all that detail. Think of it this way. These protocols and SOPs are like, as one of the videos you provided put it, written recipes, but super, super precise ones. These aren't just loose guidelines. They're like step-by-step instructions that dictate exactly how every single part of the lab work needs to be carried out. No room for interpretation.
So it's all about making sure that no matter who's doing the experiment or even where they're doing it, the process is always exactly the same, like down to the tiniest detail.
Exactly. You got it. That unwavering consistency in how things are done is the absolute bedrock of generating reliable data. You see, if you let even small variations creep into the procedures, it can throw off the results, and then it becomes really hard to be confident in what those results actually mean. For someone like you, though, who really wants to get a deep understanding, looking at these super detailed procedures as a structured approach to what can be incredibly complex tasks, well, that can actually make the whole thing less daunting. It's like having a super detailed roadmap to guide you through all those intricate steps in the scientific process.
That's a great way to look at it. Like, it's kind of like following a precise set of instructions to build, I don't know, a really complex piece of machinery. If you skip steps or try to do things out of order, the whole thing could malfunction or even, become dangerous. So what are the key building blocks, the must-haves, when you're creating these detailed blueprints for doing lab work?
Well, creating these protocols is a pretty involved process, but if you look across all the sources you've provided, a few key elements pop up again and again. First up, and it's a big one, is method validation. It's all about making absolutely sure that the tests they're using in the lab are scientifically sound and will give accurate and reliable results consistently.
So you can't just like come up with a test in the lab and start using it. There needs to be proof that it actually does what it's supposed to do.
Exactly. You hit the nail on the head. Your sources highlight several characteristics that are really important when defining a validated method. First, you need accuracy. That's how close the result you get from the test is to the real, true value of what you're actually measuring. Scientists often check this by adding a known amount of a substance to a sample. That's called fortification or spiking. They see how well the test can detect and measure that added substance. Then there's precision. That's all about how consistent the results are when you test the same sample multiple times, using the same conditions each time. It's usually expressed as something called a percentage relative standard deviation. We shorten that to percent RSD.
Got it. So accuracy is like hitting the bullseye on a target, and precision is about having all your shots land close together, even if they're not all right in the center.
Yeah, that's a great analogy. But it doesn't stop there. You also need specificity. What that means is the method should only measure the specific thing you're interested in and not be thrown off by other stuff that might be present in the sample. It shouldn't be influenced by the equipment itself either. And last but not least, there's ruggedness. This is about how well the method holds up and continues to give reliable results even when there are small, intentional changes in the experimental conditions. This might involve things like having different people run the test, using different equipment, or even doing the tests on different days. It's all about making sure the method is dependable in the real world of a busy lab.
Wow, those are some pretty strict criteria. So where do labs even get these validated methods? Do they have to invent them all from scratch?
Luckily, no. They don't have to reinvent the wheel every time. The EAS consulting group video you gave us actually mentions several well-known organizations that publish these tried and tested, fully validated methods that labs can use. For example, there's the United States Pharmacopoeia, or USP. They focus on setting standards for medicines and dietary supplements. Then there's AOAC International. They provide standards for analytical science that are accepted around the world. And the Food Chemicals Codex, or FCC, offers standards specifically for food ingredients. And let's not forget the Bacteriological Analytical Manual, BEAM, which is from the FDA. It lays out procedures for detecting those nasty pathogens in food. Labs can often just take these established methods and use them as is, or they might adapt them slightly to fit their specific needs.
Right, and it's not just about the test methods themselves, is it? The equipment used to actually perform those tests has to be in tip-top shape and giving accurate readings too, right?
You are absolutely right. And that brings us to equipment qualification. Your sources talk about this in some detail, breaking it down into different stages. There's DQ, design qualification, that's confirming that the way the equipment is designed meets all the specific requirements. Then there's IQ, installation qualification, where they double check that the equipment was installed correctly, following the manufacturer's instructions to the letter. And then there's OQ, operational qualification. That step is all about checking that the equipment actually works the way it's supposed to across its full operating range. And finally, PQ, performance qualification. This is the big one where they show that the equipment consistently performs as expected in the actual lab environment where it'll be used day in and day out.
It makes sense to have all those checks in place, especially when you consider how sensitive some of this lab equipment can be. Even something as seemingly small as a change in room temperature could potentially mess with the results.
Oh, absolutely. Even something as simple as moving a piece of equipment to a different spot in the lab could affect how it performs. Maybe it ends up closer to a vent or in spot with more vibrations. It can be that sensitive. That's why they often have to requalify equipment after any major repairs, if they've changed the equipment's configuration significantly, or if they've had to move it to a new location. And all this qualification work needs to be meticulously documented in, you guessed it, qualification protocols and reports.
And on top of making sure the equipment is qualified, there's also calibration. How does that fit in with everything else?
Good point. Calibration is about making sure that the equipment is giving you accurate and precise measurements. You see, regulations require all equipment to be calibrated before it's ever used, and then at regular intervals after that. Those intervals can be based on the manufacturer's recommendations, or the lab might set their own schedule based on how often the equipment is used and for what purpose. The main goal is to make sure those measurements stay accurate and precise over time. They'll often figure out the initial calibration settings during the performance qualification, that PQ phase we talked about. But some instruments, the ones that give you direct measurements like pH meters or those analytical balances, those might need to be checked way more often, maybe even daily, just to make sure they still want to point. And just like with qualification, all those calibration activities need to be logged in detail, including the dates, the methods used, and of course, the results.
So, if I'm understanding this right, qualification is kind of like making sure the instrument is fundamentally sound and set up the right way, while calibration is like fine-tuning it to ensure those numbers it spits out are actually correct.
Yeah, that's a really clear way to think about it. So we've talked about validating those test methods and making sure the equipment is reliable. What else is essential when you're developing these really robust GLP protocols?
I'm all ears.
Well, another crucial piece of the puzzle is risk assessment. They often call this quality risk management. It's all about identifying and evaluating potential problems, risks that could compromise the quality and integrity of the work being done in the lab. And they do this up front.
Risk-based thinking seems to be a big deal across many industries now. So how does that apply to lab protocols specifically?
Well, like those books on quality risk management you provided explained, and the ICHQB guideline too, a risk-based approach allows organizations to sort of anticipate potential issues that might pop up during lab operations. And then, even more importantly, to put preventative measures in place to minimize or completely eliminate those risks. It's about being proactive, not reactive, trying to foresee what could go wrong and then taking steps to prevent it from happening in the first place.
So instead of waiting for a mistake to happen and then scrambling to fix it, you're looking for those potential weak points in the system beforehand and trying to reinforce them.
Precisely. And they have specific tools to help with this. One is called failure mode and effects analysis. That's FMEA. They use that to systematically analyze each step in a process, looking for points where it could fail. Then they figure out how likely those failures are, how bad they could be if they did happen, and what the consequences might be. This way, labs can prioritize risks and figure out the best ways to control them. Then there's fault tree analysis, or FTA. It's more of a reactive tool used when something has already gone wrong. It's a structured way to investigate how a specific failure happened, especially if there are multiple factors that contributed to it. It helps you trace things back to the root cause.
So FMEA is like preventative maintenance, trying to stop those problems before they occur, and FTA is more like a post-mortem when something's already broken down. Got it. So is there a final piece to this puzzle, something that ensures these protocols stay effective over time?
Yes, absolutely. And that would be periodic review. These GLP protocols and SOPs aren't meant to be written in stone, never to be changed. They need to be looked at regularly and updated if necessary. That way, they stay accurate and effective, and they keep up with any changes in regulations, new technology, or even just better ways of doing things in the field.
It's like a continuous improvement cycle, constantly looking for ways to refine and improve these recipes for how lab work is done, right?
Exactly. You got it. And one of the videos you shared mentioned a really important regulatory requirement. You need to have written procedures for conducting evaluations at least once a year. And these evaluations need to cover things like complaints, any recalls that have happened, and any findings from internal investigations. This ongoing assessment helps identify areas where they might need to revise the protocols or maybe even develop brand new ones to maintain quality and stay compliant.
Okay, so we've got these incredibly detailed protocols. They've been developed with this rigorous method validation, proactive risk assessment, and they're committed to reviewing them regularly. But just having these documents sitting around isn't enough, right? People in the lab actually have to follow them and follow them consistently.
You're absolutely right. Implementation and consistent execution are critical. In good manufacturing practice, or GMP, which is closely related to GLP, there's a really important principle that says, if it wasn't documented, it didn't happen. This really highlights how essential it is to keep detailed records of everything that happens in the lab.
So seriously, every single step, every observation, every result, it all needs to be carefully recorded.
Yes, every bit of it. And it doesn't matter if it's written down on paper or logged in an electronic system. The documentation needs to be thorough, capturing all the important information, but also concise and easy to understand. And it needs to be recorded right when the activity takes place. If it's handwritten, it's got to be legible, signed by the person who did the work, and dated. And if they make a mistake, they have to correct it the right way by drawing a single line through what's wrong, initialing it, dating it, and writing a short explanation for the change. No erasing or scribbling it out so you can't read it. Electronic systems have similar rules. They often have what they call audit trails that track every change to the data, including who made the change and when.
Wow, that is a lot of record keeping. Is there any way to make sure all this documentation is accurate and complete?
Yes, there is. A really important part of making sure the data is reliable is that they have to have peer review of all the original records. That means a second qualified person looks over all the documentation, double-checking that it's accurate, complete, and follows all the established protocols. And of course, that review process itself needs to be documented too, particularly in electronic record-keeping systems.
So where does all the documentation actually live? What are the main types of records they keep in a lab that's operating under GLP?
Your sources mention a few key types. First, there are the standard operating procedures, the SOPs themselves. Those are a primary record, laying out those detailed instructions we talked about earlier. Then there are lab notebooks, which can be electronic or the traditional paper kind. They use those to document the details of specific experiments and any controlled processes they're running. And we've already talked about the equipment logbooks, which are crucial for keeping track of each piece of equipment, including any maintenance, repairs, or calibrations. Labs often use a combination of all these types of documentation to make sure they have a comprehensive record of everything they do.
So the SOPs are like the official playbook, the standard way of doing things. What happens if someone decides to go rogue and deviate from the SOP?
Well, as one of those videos pointed out, anytime someone veers off course from an established SOP, even if it seems like a minor thing, they need to follow up formally. That usually means they have to investigate to figure out why the deviation happened, assess what impact it might have had on the quality and integrity of the data, and then put corrective and preventative actions in place to ensure it doesn't happen again. The rule is stick to the SOPs. Any departure from them has to be justified and addressed. They take this stuff seriously.
It all sounds very tightly controlled, which makes sense. How does all this meticulous protocol development and implementation tie into the bigger picture of regulatory compliance?
Following those GLP protocols and SOPs is absolutely essential if they want to meet the requirements set by regulatory agencies. Your sources actually provided specific examples, like 21 CFR Part 111 Subpart J. That section lays out the specific requirements for lab operations in the world of dietary supplement manufacturing. Then you've got 21 CFR Part 58, which focuses specifically on the good laboratory practice regulations for those non-clinical lab studies.
So we're not talking about suggestions or best practices here, but actual rules and regulations that labs operating in these areas have to follow by law.
Exactly. And like we mentioned earlier, GLP and GMP, good manufacturing practices, are closely related. GLP is really focused on the lab side of things, like research and testing, while GMP covers the whole manufacturing process for things like pharmaceuticals, with the main goal of ensuring those products are high quality, safe, and effective. And one of the videos really emphasized that for any GMP activities, which would definitely include work done under GLP, the people doing those tasks have to be properly trained and demonstrate that they can do the job correctly.
It sounds like there are several layers of oversight and accountability baked into the system.
Absolutely. The quality control unit within a company plays a huge role in all of this. As they explain in 21 CFR Part 211, that unit has the final say on approving or rejecting any procedures or specifications that could affect the quality of the drug product. And their responsibilities have to be clearly spelled out in written procedures. And crucially, those written procedures need to be followed to the letter by everyone involved.
So from the very beginning, when they're designing those protocols, all the way through to putting them into practice and keeping those meticulous records of every single step, it's all geared towards guaranteeing product quality and meeting those strict standards set by the regulatory agencies. Makes sense.
Exactly. And remember, this isn't a static system. It's designed to be continuously improved. That whole cycle of developing the protocols, implementing them, and then reviewing them regularly, it's all part of an ongoing effort to refine and enhance how things are done in the lab.
And that includes when things don't quite go according to plan, like when a test gives you a result that's out of spec.
Especially then. When they get those unexpected results, the focus isn't on pointing fingers and blaming someone. It's about thoroughly investigating to understand what caused it. They want to figure out exactly what went wrong and then put things in place to prevent it from happening again in the future. One of those Season 1 transcripts you provided actually made a really good point. If you investigate them properly, failures can be incredibly valuable learning opportunities, and they can help make the whole system even stronger.
So all this incredibly detailed stuff in GLP protocols and SOPs, even though it might seem a bit overwhelming at first, it's all about building trust in the scientific data they're generating, right? And ultimately, it's about making sure that the products that can really impact people's lives are safe and high quality. So for you listening, understanding this whole framework can really help you appreciate just how much rigor and care goes into scientific research and development. It's not just people messing around in a lab.
I couldn't agree more. These protocols and procedures aren't just bureaucratic heaps to jump through. They are the fundamental tools that make it possible to generate reliable and trustworthy scientific data. And they do a lot. They ensure consistency, they help labs comply with those crucial regulations, and they drive that constant improvement that's so important in any lab environment.
So for those of you listening out there, think about this for a minute. In your own field, whatever you're working on, or even just your own interests, are there complex processes where having these detailed protocols and standardized procedures could be helpful? Could they help you be more accurate and consistent? Maybe a more structured approach could help you make fewer errors and have more confidence in the results. Something to consider.
And if you want to go even deeper, you could look into those specific regulations that might apply to your work or your interests. If you're involved in preclinical research, you could check out 21 CFR Part 58. Or if you're in the world of dietary supplements, Part 111 is the one to look at. And for a broader international perspective on those quality standards in the pharmaceutical industry, you could look into those ICH guidelines from the International Council for Harmonization. Lots of good stuff out there.
And if you've got any feedback or any other questions you'd like us to explore, let us know. We could always do another deep dive focused on specific parts of GLP or GMP, getting into even more detail. Thanks so much for joining us today. We had a great time digging into this fascinating world of lab protocols and procedures with you. Until next time.